Hello, good morning, everyone. Thank you for being on time. Let's get started with the test one review. So I'm just gonna go through the questions in the review package. And if there's anything you want me to elaborate on, please do let me know. The test, of course, is on Wednesday, and there will be no class on Wednesday. You just have to do the test, and that's it. And it covers everything from lecture 1 to lecture 5. So let's get started with the review. First question, what is a placebo? Give an example of an experiment where a placebo is needed. Give another example where a placebo is not needed. So a placebo is essentially um, a dummy treatment, right? A sham treatment, a treatment that has no effect on the results that you're looking for. So placebo has no effects on the outcome. The purpose of a placebo is to minimize bias, right? So an example of where you need a placebo is when you are dealing with human subjects. Okay, so needed for testing a new drug. Right, so you would have three groups. You have the control group, you have the treatment group, and then you have the placebo group. Okay, control group receives nothing. The treated group receives the experimental drug, and the placebo is going to receive a dummy drug. When do you not need a placebo? You don't need a placebo for a lot of things. For example, animal testing with animals, for example. You don't need a placebo for like the beetroot experiment that you'll be doing. Okay, because there's no bias in those situations, right? That needs to be eliminated. All right. What is a valid hypothesis? For a hypothesis to be valid, it must be testable and falsifiable. You must be able to conduct an experiment to check whether it is correct or not, and then it must be potentially wrong. There will be no written questions for the upcoming test, so you don't really have to practice writing the hypothesis, but you should practice writing the hypothesis for your lab, your Beetroot lab, which is going to happen next week when Professor Moran takes over. He will be explaining to you what needs to be done. But one of the things you need to do is to be able to write a hypothesis. All right, next question here. We have two graphs, and it's asking you to take a look at whether or not the new drug is effective. I'm just trying to make these a little bit bigger. There we go. So the first one, the N number tells you how many people are involved in the study. Generally speaking, the bigger the sample size, the more people you have, you include in your study, the more accurate the results will turn out to be. So we have the control group here, and 70% of the people experience jet lag. When you give them the placebo, only 40% reports symptoms of jet lag. And then when you give them the experimental drug, also roughly about 40% of the people experience jet lag. So based on this result, is the drug effective or not? This is something we talked about before, but does anybody want to give it a shot? Is the drug effective or not? Why? Yeah, go ahead, Tosh, please.
You're talking about the left chart, right?
Yep, this one on the left.
Yeah, based on the left chart, I believe it is effective. Because there's a significant difference between the placebo, the amount of people that receive the amount of people that um, have benefits from the placebo and the amount of people that have benefits from the actual drug.
Okay, yeah, thank you
So
for
I'd
your.
say it is effective.
All right. Anyone else want to give this a try?
Vincent, are you talking about the one that you say called the N,
This one,
right?
yeah, this one. This is the one I'm looking at. Yes. Do you think based on this data? Yeah, go
I
ahead.
don't think based on that data, I think that shows that maybe the placebo group and the control group both are experiencing the same thing. So I think the pill doesn't work because it's showing that they both have reduced maybe symptoms of jet lag.
That's right. Thank you. So this is ineffective. The drug is ineffective because both placebo and treated group as in like the actual drug, okay? Both placebo and the treated group were able to reduce symptoms, right? When compared to the control, right? It shouldn't be this way because remember the placebo is not supposed to do anything for you, right? So when you see a result like this, right? This suggests jet lag is all psychological, right? Well, we wouldn't say it's all psychological. It's partly, right? Psychological, because you give these people some pill, you tell them we're gonna help them sleep better, and then they turn out sleeping better, even though the pill is not, physically doing anything for them, right? So the fact that both placebo and the treatment group were able to reduce the percent of people who experience jet lag, this tells you the drug is ineffective. On the other hand, over here, this is effective. This is when the drug is effective because only the treatment group is able to reduce the symptoms of jet lag. Okay, placebo has the same outcome as the control group. Okay, I hope that makes sense to you. Any questions before we move on to the next one? All right, so this is not like an exhaustive list of things that you have to know. These are just some of the things that jumps out to me. Things that people tend to get confused about. So make sure you go through these. But then there are also other things like the experiments, right? The Rady experiment, the one with Pasteur, right? The one with the flies, right? We talk about those experiments, and the one with the broth and the swan neck and all those things, right? Those are also fair things to ask on the test. All right, let's take a look at this next part here. This is lecture 2. What holds atoms together in a water molecule? Water molecule is a covalent compound. And we know that because it is H2O. It's not a metal plus a non-metal. It is all non-metal. So therefore, what holds them together? That would be, as I said, a covalent bond. Okay. In a covalent bond, electrons are shared as opposed to transfer. How would you describe a water molecule? I would say a water molecule is polar which means it has uneven distribution of electron. Okay, mind you, the oxygen is slightly negative, right? Compared to the hydrogen. There is a question in the chat. Is there a study guide we can go over on eCentennial? This is it. This is the study guide for the test. That's on the eCentennial for the test, right? It's did you know where to find this, Takiri? No? All right. Hold on a second. So you guys gotta watch the announcement, right? Announcement. And there's a little like a bell on top of the eCentennial page. Every time I post something, the icon will have an orange dot on it, right? That's when you can check it. So for those of you who don't have it, I just post the link in the chat box. Moving on. So the molecule is polar, but it's neutral. Okay. Overall, the molecule is neutral because there's no extra electron or missing electron. It's just that the distribution of these electrons are uneven, which makes it polar. So what that means is when I have one water molecule next to another water molecule, they will be attracted to each other, right? The slightly negative oxygen will be attracted to the slightly positive hydrogen of another molecule. And this interaction between them, this is called hydrogen bond. Hydrogen bond. Okay? Just stop me if you need me to elaborate on any of these. Next question. If you supply radioactive nitrogen atoms, that just means it's something that you can trace and detect later on. If you supply that to a cell, what molecules would you expect to find the radioactive labels in? This is just another way of asking you what molecules contains nitrogen. So tell me, guys, what molecule contains nitrogen? Molecules in a cell. Right? Yes, Stella?
Protein.
Protein, yeah, there you go. Protein certainly contains nitrogen. Okay. It contains C, H, O, and N, right? And trace of sulfur, actually. What else? What else contains nitrogen?
Citric acid.
Citric acid. Actually, no. Good try. Citric acid is actually just carbon, hydrogen, oxygen. It's not something we talked about though, so like you wouldn't know that actually. But it doesn't contain nitrogen. I'm thinking about like the macromolecules, right? The 4 macromolecules that we learned. What's another one that contains nitrogen besides protein?
DNA.
Yeah, nucleic acid, right? Not just DNA. Nucleic acid contains DNA and RNA, right? So DNA plus RNA. Both of them would have nitrogen in it, right? Carbon, hydrogen, oxygen, nitrogen, and phosphorus. Okay, so you would expect to find a radioactive label in these molecules. Okay, regarding DNA, what is the monomer of DNA? That would be a nucleotide. And a nucleotide has 3 parts to it. There is the sugar, there is the phosphate, and then there is a nitrogenous base, which can be A, C, T, or G. Okay, so that's a nucleotide. And when you join many nucleotides together, you end up getting the DNA, right? How are nitrogenous base classified? So we have the purine, which is adenine and guanine. And the phrase you can use to remember this is, pure as gold. Purine is adenine and guanine. These have 2 rings in it. Okay, 2 rings. And then you have the pyrimidine. Pyrimidine, which is cytosine and thymine. This is 1 ring. So as far as the base pairing rule goes, adenine pairs with T, and cytosine pairs with G all the time. Right? So if you know one strand of DNA, you'll be able to predict the other strand. So example, if I have A, T, C, C, G, T, we can predict what's on the other side. Remember, DNA has 2 strands, right? Double-stranded. So what does what does A pair with? A pairs with T. What does T pair with? A. C will go with G. C will go with G again. And G goes with C. T goes with A. So there we go. If you know one end, you can predict the other end.
Sorry to interrupt you, Vincent.
No problem. Go ahead.
So this A and T, C and G.
Mhm.
So the top would be the first half of the DNA strand,
Yeah, this is one strand. Yeah,
and
And
the
then-
second.
this is the other strand, and then they would twist together to form the double helix.
Okay. Okay.
What
Thank
force?
you.
Yeah, no problem. What force holds the nitrogen bases together along the length of the DNA molecule? In other words, what's holding the A and the T and the C and the G? That turns out to be hydrogen bond. Hydrogen bond. So there are 3 hydrogen bonds holding G and C together. And then 2 hydrogen bonds. 2 hydrogen bonds holding A and T together, which means DNA with high percent of C and G are harder to pull apart. Okay. And remember, if you need to listen to the lectures again, there are recordings on eCentennial, right? That you can access to refresh your memory. Okay, next, what is the significance of base pairing rule? This allows you to predict, to create, I can say. It allows you to create the complementary strand of DNA. Okay, so if you have one strand, you can always figure out what's on the other side. Okay, and also, I don't know if you noticed this, but the G and the C, they are over here, right? The G and the C, G has 2 rings and the C has 1 ring. A has 2 rings and T has 1 ring, right? So across the width of the DNA, it's always going to be 3 rings wide. So this base pairing rule ensures constant width along the DNA strand. Okay, so these are some of the things that you should be aware of. Okay. Moving on. What is dehydration synthesis? Illustrate with the diagram, right? So we've done this before, right? Dehydration synthesis is, you know, removing water to connect two monomers together. Okay, so like if I have a hexagon, which represent a glucose in some cases, and then I have a pentagon, right, which represent a fructose in some case, I can remove an H2O here, right, and form a bond. Right. That's dehydration synthesis. You can repeat this process over and over again, and then you would end up having a long chain. Okay. Any questions so far? Okay, next one. Regarding enzymes, what is an enzyme? Enzyme is which macromolecules form enzyme? Does anybody know?
Protein.
Protein, that's right. Thank you. That is the protein. And how does the enzyme work? Enzyme works by lowering activation energy, right? Lowering activation energy. That's the energy required to start a chemical reaction. Okay, so sometimes the energy requirement is too high. It's not possible to achieve that within the body, so the enzyme lower that requirement, and you're still able to do it. What factors affect the functionality of an enzyme? Because an enzyme is a protein, so anything that changes the shape of the protein is going to affect how the enzyme works. So things like temperature, right? pH, salt concentration, right? Hydrophobic environment, right? Proteins are supposed to be in an aqueous environment, hydrophilic environment in the body, but if you change it to a hydrophobic environment, it's totally not going to work. Okay, temperature will heat up the protein. It will cause them to denature, right? Denature means it unravels, and then it might not work anymore. What does the enzyme lactase do? ASE often means it is an enzyme. And in this case, lactase breaks down lactose. Right? Lactose is going to become glucose plus fructose. Sorry, galactose. Right? And you need an enzyme to do that, lactase. Right? Complete the following chart. All right, so we have a bunch of questions here related to you know, all the macromolecules. So let's just go through them one by one. We start with the carbohydrates. So monomers of carbohydrates are monosaccharide, right? Monosaccharides, which include,
Glucose,
yep, thank you, there's glucose. What else?
fructose,
Yep. And just one more, please. Anyone?
lactose.
And that
Galactose, yeah, galactose. Okay, so these can come together, right, to form the disaccharide. Glucose plus glucose gives you maltose. Glucose plus fructose gives you sucrose, and then glucose plus galactose gives you lactose, right? All these are in your notes, but this is only asking for the monomers. So biological function, what does it do? So for carbohydrates, right, we have things like glucose is used for cellular respiration. I'm just gonna merge it with the example, okay? Cellular respiration. Okay, and then another biological function, we have glycogen. Glycogen is the storage form of energy in animals. We have cellulose, right, is the structural component of cell wall, for example. And does anyone have another one? Like at least two more we can say here. If all these sound very unfamiliar to you, you need to make study notes to prepare for the test. Okay, you need to go back to the earlier study guides and go through them. Anyone? Two more. What's the storage form of energy in plants called? It's not glycogen. What is it?
Starch,
Starch. Yeah. Starch is the storage form of energy in plants. And then there's also something. Yeah, go ahead, Fatuma.
Are we giving functions for each monomer or?
No, these are not monomers, right? The glycogen, the cellulose, and the starch, they are actually polymers. They are polysaccharides. But I'm just naming any kind of carbohydrates that you remember, right? That we talk about in class that has a biological function.
Okay, thank
Yeah.
you.
One more, guys. Anyone?
Chitin.
Yeah, that's right. What does that do?
structural support.
That's right.
Yeah,
Forms
hard parts of small animals.
excellent, right? Exoskeleton, right? We call them, right? Like the shell of a crab, right? The crunchy part of an insect, all that stuff. Okay, that is that. I will of course post this note after, so if you didn't have a chance to copy everything, that's okay as well. Monomers for protein is amino acid. There are 20 different amino acid, and they are all very similar. Okay, so they all have a central carbon. We'll say 20 amino acids. Okay, they all have a. This is called a carboxyl group. And yes, we actually did learn all these things. There is an amino group, and then there is a side chain. So what makes the 20 amino acids different from each other is the side chain, right? Some are acidic, some are basic, some are hydrophobic, some are hydrophilic. so on and so forth, right? So different side chains is going to give the amino acid different properties. Okay, we can connect amino acids together to form a peptide bond, right? And then it becomes a polypeptide, a protein molecule. Biological functions, there are many. It can form hormones, right? So example of a protein hormone would be insulin. Yes, Tash, go ahead.
Sorry to go back, but what's the biological function for galactose?
There's no, we didn't talk about that. Okay, galactose is going to combine with glucose to form lactose. Okay, so maybe provides energy, but these are the main ones that we talked about. Okay.
Okay.
If I didn't talk about specific ones like lactose or fructose or whatever, then you're not responsible for it. Okay.
Okay.
All right. So hormones, insulin, another function of protein. Yeah.
Enzymes.
Enzymes. Yes. Enzymes will help catalyze reaction, right? Catalyze reaction, lower the activation energy. You can really make up any enzyme, but you know, I'm just going to use the lactase as an example. We learn about proteins, membrane proteins specifically, in what was it? Lecture 4, I think. Lecture 4, right? So membrane proteins. There are lots of functions associated with membrane proteins. I'm not going to list them here, but to refresh your memory, they are involved in cell signaling. They are able to form junctions. They can attach to cytoskeleton, right? They can act as receptors, so on and so forth, right? Transport. Right, so all these are proteins function. Also, there is a structural support, e.g., like hair. Well, I guess not really hair, but like fingernails, right? Nails, muscle. All these are proteins. Anything else you guys want to ask me about proteins? Do you guys have another test this week besides mine?
Yeah, we did.
Chemistry midterm.
Chemistry? Yeah. How about math?
Chemistry.
Did you guys finish math already?
No, not yet.
Not
Yes.
yet.
I think that's
Oh,
next week.
next week. I see. Yep. I know. It's a lot of stuff. Okay, lipids. Moving on. Lipids, we have monomers. It's not really monomers, right? For lipids, it is just components, right? So we have things like glycerol. You can combine it with fatty acids, saturated, unsaturated, doesn't really matter. Both are acceptable. You can have like a phosphate group for things like phospholipids. You can have carbon rings for steroid, right? So triglyceride, that is for energy storage, right? Insulation keeps us warm. That's fat, right? Basically. We have phospholipid. Phospholipids is the major component of cell membrane, right? Does anybody remember what the carbon rings form? I have 4 carbon rings that are fused together. What kind of lipid does that give you? That's okay. Steroids, right? Steroids are fused carbon rings and they can form hormones such as estrogen, or they could be cholesterol, right? And we should know by now, cholesterol helps maintain the fluidity of the cell membrane, right? Right, so these are some functions of lipids. Okay.
Sorry, Vincent, can you please explain the fat insulation again?
Yeah, so like around major organs, right? Like the heart, the liver, the kidneys, they all have a little bit of fat that surrounds them, and it keeps it warm. Right? When it's -40 outside, your internal organs are not gonna be cold because the fat helps insulate, right? Fat is an excellent insulator. Is that okay?
Yeah, thank you.
Yep. Okay, then we have the nucleic acid, right? The monomer for nucleic acid, I just told you, is nucleotides. It has a phosphate, a sugar, and a nitrogen base, right? Nitrogenous base. We've only learned 2 types of nucleic acid, DNA and RNA. And you will learn more about it in the second half of the course, but DNA contains instruction, right? They are the instruction to make protein. Whereas the RNA, it is a copy of DNA. Remember we talked about from transcription, from DNA to RNA is transcription, right? I can write it out for you. Excuse me. So from DNA to RNA, you can do transcription. That's making a copy of DNA in the form of RNA. We don't want to take the whole RNA, whole DNA out of the nucleus. It's too big, and we can damage it, right? So you only copy what you need. That is the transcription process. Once you have it, it will go out to the cytoplasm. The ribosome will come, and then we can do translation. That converts it into protein. It reads the instruction in the RNA to create a protein. Okay. So that's that. All right, I'm just gonna let you catch up with some of these things. Okay, leave it like that. I will be right back. Give me a couple minutes, okay? Just copy what you need. Sorry about that. So any questions about anything before we go on? Okay. How is the shape and the structure of a protein stabilized? So there are 4 levels, right? There is the primary, secondary, tertiary, and quaternary, right? So primary is just the covalent bond holding the amino acid together, right? We can say peptide bond actually. That's the specific kind that holds them together. For secondary, you might remember there is the alpha helix and beta sheets, right? Okay, that's the coil, and then the zigzag that holds the backbone together. And then for tertiary, we have different forces. These are interaction of side chains. Right, so we have like ionic interaction, we have hydrophobic interaction, right? Hydrogen bonds. These are all in the PowerPoint slides. Okay, there are a few slides that goes through this concept. And it shows you what holds the protein shape together. As you know, the shape of the protein is directly related to its function. So if you alter the shape, you will alter its function. In many cases, you will lose the function completely and it will not work, right? So there is a key and lock relationship between the shape and its function. And quaternary, it's not always there. This only happens when you need to have multiple proteins coming together to form a complex, we call it. So for example, like in hemoglobin, right, we would have 4 different proteins that come together and it forms a complex. So which proteins do we need to bring together? That's the quaternary structure. And what holds it together would be interaction between polypeptide. Okay, so that's a polypeptide. Polypeptide is essentially referring to the protein itself. Okay, so these are like 4 different subunits that are going to be glued together by these interactions. Okay, so when you heat things up, right, when you heat the protein up, you will disrupt the quaternary structure, tertiary structure, and secondary structure, but peptide bond will stay intact. The primary structure is the strongest. Okay, this is the strongest one. The strongest. Hard to break. Okay, you can't just heat it up and break the primary structure. But you can break all these other structures. So heat will destroy the shape of the protein, but the sequence will usually remain intact. Okay, you might want to know how to identify these things. The first one here, hopefully you recognize it after all that we've talked about. That is
Protein.
it will form the
Amino
protein.
acid.
That's right, this is an amino acid. How about this one here? Does anybody know what this is? There is the phosphate. There is the sugar. And there is the 2 rings here. So some kind of nitrogen base, because it has 2 rings, is a purine. So what is this whole thing?
Nucleic acid.
Yeah, nucleotide. Right? Nucleotide, which forms the nucleic acid. Glucose here. And this is just a fatty acid. Okay. Trish, you have a question? I see you unmute your mic.
No, I was just asking you what the polypeptides were.
Oh, so like, you know, when the amino acid links together, right? This whole thing is called a polypeptide. And then the polypeptide will fold up, and then this forms the protein. So essentially polypeptide is just the whole sequence, right? Yeah,
Okay.
yeah, it's like saying polysaccharide, right? Polysaccharide is like all the glucose linked together, right?
Yes. Okay.
All right, so as promised, last class, I went through the test paper, and then I found some questions that people didn't really quite get correctly. So I'm gonna go through them with you. Let's go through the ones for the first two from quiz 1 first, okay, which is lecture 1 and lecture 2. Okay, so these are questions where yeah, like a lot of people got them wrong. Okay, like you might not recognize them because it might not necessarily be on your quiz, but nevertheless it might still show up on the test, right? So here we go. Peer-reviewed journals are generally more reliable sources of information because, so just to remind you what peer review is, peer review means, you know, when people do a scientific study and they want to publish their results, they need to, if they want to do it in a peer-reviewed journal, they need to submit their work to other experts in the field so that they could examine the work and make sure that it was done with the proper standards and, you know, identify any potential errors, and then they have to fix those things before it's actually published, right? So they are generally more reliable because the findings from experiments are scrutinized by experts in the field for accuracy, appropriate experimental methods, and interpretations before publication. That's the reason. Okay, that's the reason. Let's take a look at the other ones. The process immediately validates any hypothesis tested in the experiment. That's not true. Primary means for information other scientists of new information in the field. No, p-hacking does not occur. So just a quick word about p-hacking, right? I think people also not quite get that. P-hacking is just subconscious or conscious manipulation of the data, right? So that it achieves the result that you want. Just because it's peer review doesn't mean p-hacking doesn't occur, right? In fact, some people have been caught doing p-hacking on large scale studies of things that are not true in the past, right? And these are published in very prestigious journals. So that certainly does occur. Some are funded by government, some are not. It doesn't really matter. But so like the main one for the peer review journal is this one, that there are other experts in the field that will help scrutinize your work. Now this one was like, I think it was on everybody's quiz. I deliberately put this on everybody's quiz, but only like 20 to 30% of people got it right. And this was the question that was from the PowerPoint slides. Remember we do those poll questions during class when we go through the PowerPoint slides. You should also study those. Right? Some of those are going to show up on the test. Right? So this one, it's asking for deductive reasoning, which is going from general to specific. And the example is this, the answer is this one. All virus requires a host to replicate. And then COVID-19 is a virus, it must require a host to replicate. You apply a general statement to a specific case, right? It's not the first one, even though it sounds very similar. This one is backwards, right? Remember in class, I gave you another examples with wings and bees and birds, and the conclusion was like, bees are birds, right? That's this one. This is the wrong one, right? So if you need a refresher on that, please go back to the lecture 1 recording, and it should be towards the end, the last 5-10 minutes of the recording. You will hear the full explanation of this. Okay, but this is the answer. The point I'm trying to make is go through the PowerPoint slides, find all those poll questions that we did, and make sure that you know the answers to those. And I can hear some of you saying, but those questions, some of them are too easy, right? Even if I know how to do that, it's not really going to help me on the test. But think of it this way. If you cannot even do those questions, then there is no chance you will do well on the actual test question, right? So go through the poll questions, make sure you at least know how to do those before you move on to your studying. Okay. The formation of polymers involves a process called what? Okay, we warning, yeah, we did this already, right? This is the dehydration synthesis, right? We remove a water molecule to connect two monomers together so that you would get the polymer. This one, let's do a poll, right? See if you guys are there. Now, let me just copy this thing over to help you. There. That's the amino acid that we drew. Okay, I don't know why I cannot put it here. It's like doing weird stuff, but there we go. Okay. So compare it, right? What is not in the amino acid? Okay, let's see. Carbon atom, check, it's there. R group, that's the side chain, check. Peptide bond, I don't see any peptide bond here. That probably is the answer, but let's just be sure. An amino group, okay, check. Hydrogen atom, check. Carboxyl group, check. Oh, must be peptide bond, right? A peptide bond is what connects two amino acids together, right? When you want to create a polypeptide, right? Okay, it's not within an amino acid, right? So that's what I mean, right? Like, I know I said this before, but I'll say it again. It's important if you have trouble remembering everything, you have to create a help sheet that's useful on the test, right? If this is very difficult for you to remember, put it down on your help sheet, and your help sheet should be organized by lecture. You should have a help sheet for each lecture. Okay, these are not like your full notes. It's not something for you to like, you know, search through during the test. These are concepts that you just, things that you can't remember. Okay, maybe you have trouble memorizing all the polysaccharides, what they are, right? So then, you know, if you see a questions about polysaccharides, at least you will know where to look for on your cheat sheet, rather than, you know, scrolling through your PowerPoints and trying to find the answer, right? Things like that. I hope that helps. Okay, this is another one. Which of the following is not true regarding macromolecules? Okay, so an example of macromolecule is proteins. That's definitely true. There are 4 macromolecules, right? Carbohydrates, proteins, nucleic acids, and lipids, right? So that's correct. We're looking for the one that's not true. Cells produce macromolecules by the process of hydrolysis. Okay, how do we build macromolecules? We actually just had another question earlier, right? We make macromolecules through dehydration synthesis, right? Hydrolysis is to break them. So this is not true. It should be dehydration synthesis. Okay. When you reject an answer, when you choose a particular answer, make sure you have a good reason for it. Don't just choose it because it feels right. Choose it because you are able to correct the statement, right? I know exactly what's wrong with this. Therefore, I'm confident this is the result. This is the right answer. Okay. You can take a look at the other ones as well. Cell uses macromolecules to signal other cells. Now that sounds a little bit abstract. You might not remember learning about it, but you are supposed to know, well, macromolecules include proteins. And we know proteins can be used to signal other cells, right? Cell signaling is one of the functions of proteins, right? When it's in a membrane. Cell uses certain macromolecules to store energy. Glycogen is a macromolecule, starch is a macromolecule, and we know they are used for storing energy in animals and in plants, right? The subunit needed to make macromolecules important to human bodies results from digestions of food, right? How do we get all those subunits? How do we get those monomers? We get it from our food, right? Remember there's a diagram with the food, and then you break it down into monomers, and then you use anabolism to build them, right? So the one that's not true is B. All right, moving on. Do you guys recognize some of these questions? Like maybe it was on the test. Like, yeah, if you don't recognize them, either you really don't recognize them when they were actually on your test, or I mean, at least some of them must be on your test. Okay, it's very unlikely that none of these was on your test. Anyway, I don't know what I'm talking about anymore. A research student is analyzing the nucleic acid of a virus. He finds that nucleic acid contains thymine. From this, it can be concluded that the nucleic acid, and then blah, 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 blah. This would be an example of an application question. It's not like straightforward. You need to think about it, right? This would also be, some kind of application question, right? So it contains thymine. Is it a strand of RNA? No, it's not, because there are no thymine on RNA, right? RNA contains uracil, no thymine on it. So if anything, it should not be RNA. Contains glucose. Glucose does not even have anything to do with nucleic acid. That's not the right answer. Contains ribose. Well, if thymine is only found in DNA, right? Does DNA have ribose? No. DNA has deoxyribose. That's what the D stands for, right? It is actually a protein. No, thymine has nothing to do with protein. Protein is amino acid. It is double-stranded. That's the right one. That's the right one, because DNA is double-stranded, right? Do you guys understand the logic here? I certainly hope so.
So it's not a,
Could you
No,
please
no,
repeat that?
yeah, just because it contains thymine doesn't actually tell you, it's protein. In fact, there's no thymine in protein, right? Protein is made up of amino acids, right? Thymine is a nitrogenous base. Okay. This questions I did in the lecture, and then I did it again during the tutorial recordings.
I thought because it came under nucleic acid, it would be considered a form of protein.
Yeah, but nucleic acid is not protein, right?
But
there are four
Isn't
kinds of
protein what makes up the DNA?
No. DNA is made up of nucleotide, right?
Oh,
Yeah, there are four kinds of macromolecules, right? Carbohydrates, lipids, protein, and nucleic acid. Protein and nucleic acid are two separate categories, right? Yeah. I'm glad we got that sorted out. Next, a certain species of animal, 33% of its base is A. What is percent of C? If you have 33% adenine, you also must have 33% of what? Which other base is also 33%? Help me out here, guys.
Thymine.
That's right, because A pairs with T, right? So whatever percentage of A you have the same percentage of T. So if you add them up, 33 plus 33 is 66%. Okay? So if I take 100%, which is everything, minus 66%, I will get 34%. 34% will be C and G together. Right? 66% makes up A and T. The rest must be C and G. And they must be the same amount, right? C pairs with G. So 34 divided by 2 gives you 17. All right,
Can you do that again, Vincent?
sure. Do you understand why I put 33 for T?
Because you're saying it should be similar.
It should be the same. It should be the same as A, right? Because
Oh, okay.
A pairs with T, right? Remember earlier in the review, A pairs with T and C pairs with G, right? So whatever you see percent you see for A, it's the same percent for T. So I add them up, 33% plus 33% gives me 66%. If I take the total amount of genetic material, all of them 100%, I subtract the 66, I subtract the A and the T, all that remains would be the C's and the G's, right? So 34% makes up the C's and the G. But the percent of C equals to the percent of G. So I take 34, breaks it into 2 equal parts. I get 17%. Okay. The difference between, oh, so this is like the quiz 2 now. All right, so we'll come back to it. Any question about lecture 1, lecture 2 before we move on to 3 and 4? Okay. Now that we have more people, I just want to say one more time. There's no class on Wednesday. Right? You don't have to come to Zoom. Right? You just study more in the morning, maybe, and then you take the test, right? The test will be available from 6 a.m. to 11:59 p.m. like usual. All right, next. List the major components of cell membrane. We drew this out. Okay, so I'm gonna say refer to the fluid mosaic model. Okay, we drew it in lecture 3. We probably still have that picture. Oh, sorry, lecture 4. I mean, is it lecture 4? Yeah, there. This is the fluid mosaic model, right? And I'm just copy it over for you. That's the one. Okay, so different components of the cell membrane. We have the phospholipids, we have the cholesterol, all these things. Make sure you know what each one of them does. And if you're confused, let me know. Okay, email me. Central dogma of biology. Right, we talked about that earlier already. We have the transcription. That's the DNA being copied in the form of RNA. So let's write that down again. Transcription. And this happens in the nucleus. And then the RNA will exit through the nuclear pores. It will go into the cytoplasm. And it is in the cytoplasm that the RNA will be decoded by the ribosome to make protein. This is translation. Okay.
This is how you want us to answer?
For what? For 10?
This question, yes.
Yeah, that's the central dogma, right?
Okay.
But again, there's no written response on the test, right? Okay, so these are just concepts that you should be familiar with in case it pops up in the question. So for translation, ribosome decodes RNA to make protein. This could happen either using ribosomes that are freely floating on their own, or it could happen in conjunction with the rough endoplasmic reticulum, right? Because ribosome can be by themselves, or they could be attached to an ER. So the difference is in the ER, those proteins will be packaged into a vesicle. and that vesicle will move on to other parts of the endomembrane system. If it's a protein that's made by ribosomes that are freely floating, no vesicles for those. One of the functions of liver is to remove toxin from blood. Based on this function, which two organelles would you expect to be abundant in liver cells? Okay, so detoxification, right? So there are a bunch that would play a role in detoxification, like the lysosome, right? Smooth ER also plays a role in detoxification. And then there's the peroxisome. I know it only said list two, right? Now there is more to this question. Right? This is just an example of one way I could ask you this, right? You know, I could change it to like one function of muscle is to provide constant contraction. What organelles would be abundant? Right? That would be mitochondria, right? I could change it to one function of the adrenal gland, which you have never heard of, but I can tell you that the adrenal gland makes steroid hormones. Based on that, what organelle is going to be abundant, right? So you see, you basically have to know what organelle does what, and then relay it to a particular description I give you, right? So I was saying, right? I don't know if you caught the whole thing. I was saying, e.g., adrenal gland makes steroid hormone. Okay. So which organelle would be abundant in the adrenal gland cells? Which organelles would you have a lot of? What do you guys think?
Lipids?
I'm sorry?
Lipids?
Yep, there's something to do with lipids, but lipid is not an organelle, right? Lipid is a macromolecule. The question is, what organelle would you have a lot of? But you're on the right track. Yeah, for those of you a little bit confused, right? Listen.
Maybe the SCR.
Exactly, right? Exactly, right? So what is this question really asking? This question has nothing to do with the adrenal gland. You don't even have to know what the adrenal gland is, right? So like for some people, they might panic and then they quickly pull out their phone and search up adrenal gland. That's like not gonna help them at all, right? What this question really is asking you, basically, is asking you which organelle makes steroid hormone. That's what this question is about, right?
The you said it, the smooth endo
Yeah,
plasma
exactly that one.
reticulum.
Yeah, I didn't say it. Some other student said it, right? And
Oh,
that's the
you
correct
wrote it.
answer.
Oh.
Yeah, SCR, right? Smooth ER, right? Yeah, no, this was for like detoxification, right? So I'm saying, you know, if something makes steroid hormone, steroid is a lipid, and lipids are made by the smooth endoplasmic reticulum, right? Okay. So there are different versions of this question I can ask you. Right? If I say, oh, this one makes a lot of protein hormones, right? Then you would choose, you know, rough ER, right? That's where proteins are being made. Do you guys understand? Yes, go ahead, Tash.
Sorry, I was just wondering, for the test, do you know how many questions will be short answer and how many questions will be multiple choice?
Everything is posted on eCentennial. If you haven't had a chance to look at that, please do so. And I just want to say one more time to everybody, there's no short answer on this test. Okay. This test is, oh, did I not post that information? Oh, I guess I
No,
did
no.
not. Oh, sorry. I thought I did. Hold on, give me a second. Oh, I really thought I did. Let me see. So I am looking at it, let me give my calculator. Mm. Hold on, okay. So there are 8 multi-select question, 8 multi-select question. Okay, those are the questions that have more than one correct answer. And let me see, for the application question, it is 15 application question. Okay, so 8 multi-select. These are questions with, you have to check all the correct answers, right? What did I say? 15 application questions. And then the rest of them, the rest of it would be regular question, which is 20. No, that doesn't seem right. Let me see. 27 multiple choice. Okay,
Hi,
so
Professor. Please, could you remind me what application question means?
like this one, right? Like I give you a scenario and then you have to figure out the rest, right? Let me see. Like something like this, right? This would be an application question, right? Instead of asking you, thymine is found in, and then I give you like, you know, carbohydrates, protein, DNA, RNA, lipids. That would be straightforward, right? So that's regular question. But this one, I give you a scenario. You have to apply it in a situation. Okay. Don't get too hung up on whether something is application or not. It would appear the same to you. Okay, like it's not going to be in a different section. It's all going to be mixed up, right? Okay, so when you see a question, don't try to think whether it's application or regular question. It makes no difference. You just do it the same way, right? Are we okay? I guess the best way to tell you is like applications are not questions you can just Google the answer for. Okay, like it's not something that you will be able to find the answer readily. You really have to think about it, right? So that just levels the advantage a little bit for online assessment. Any other questions before we continue with this review? All right, and Tosh, thank you for bringing the fact that I didn't have this posted, bringing it to my attention.
No problem. Thank you so much.
Yeah. Describe the structure of a mitochondrion and state the function of each part. We did that before, but now, you know, let's just review a few extra things that we know. So there is the mitochondria. We have the outer membrane. All right, we have the inner membrane, which forms the cristae, right? Okay, so remember, in the matrix, that's where pyruvate oxidation and Krebs cycle happens. And then embedded in the cristae, that's where you will have the electron transport chain, right? So when you're studying, you should try to connect different units together. Okay, that is the mitochondria right there. Okay. Describe the endomembrane system. That is certainly something that you should review yourself. Very briefly, we will have the rough ER. and then you will have a vesicle, right? The vesicle is going to go to the Golgi body. And from the Golgi body, it might become a lysosome. Right? It might become a vesicle that are storing protein, stored in cytoplasm until you need it. You might release the protein out of the cell, right? Exocytosis, right? It might become a membrane protein. And then we also have the SER, which is going to produce lipids. Okay, so the lipids could be like a steroid hormone, right? It might become part of the membrane, right? Because if you're making phospholipids, for example, if you're making cholesterol, all that stuff is going to go to the membrane, right? So this is just a very condensed summary of the endomembrane system. For the detailed explanation, right, look at lecture 3. All right, let's take a look at some more questions. Well, let's just go through them, even though some of them overlaps with lecture 4. So these are from quiz 2, right? The difference between pinocytosis and receptor-mediated endocytosis is that, for those of you who forgot, bulk transport, which is a type of active transport, it requires ATP. It transports things against the concentration gradient. There are different kinds of bulk transport, right? We have exocytosis, that's sending things out of the cell. And then we have endocytosis, that's bringing things into the cell. And in terms of endocytosis, there are 3 kinds. There is phagocytosis. Excuse me, phagocytosis, which is to bring in solid particles. We have pinocytosis, which is to bring in liquid primarily. And then we have the receptor mediated endocytosis. Okay. So I think that's what you guys remember. I remember some of you said last week that this the questions are very wordy and sometimes it takes some time to read a couple of times before you understand it. Maybe this is one of the example, right, of those questions. So let's see. We are looking for the difference between pinocytosis and receptor-mediated endocytosis. First, pinocytosis can concentrate substances from extracellular fluid. That just means the outside environment. But receptor-mediated endocytosis cannot. Well, both of them are bringing things in, right? Okay, so it's not about concentrating things or not concentrating things. You're not changing the concentration of something here. So that's not the answer. Pinocytosis is non-selective in the molecule it brings into the cell, whereas receptor-mediated endocytosis offers more selectivity. That's the right answer. In fact, in the lecture notes, I told you receptor-mediated endocytosis requires whatever you're trying to bring in to bind to a receptor, right? You might remember the picture goes like this, right? There are some like Y-shaped stuff here. And then I think there were some like orange circles, right? So you can't just randomly bring them in. They must fit the receptor. If it doesn't fit the receptor, you're not going to bring them in. That's the whole point of receptor-mediated endocytosis. It offers some kind of selectivity. Okay? It allows you to choose what to bring in. Whereas pinocytosis, it's, you just bring in liquid without being too picky about it. Okay? Pinocytosis require energy, but the other does not. That's not true. Both of them, as I told you, are active transport. They both require ATP. Pinocytosis only brings in water, but receptor-mediated endocytosis bring in other molecule as well. So you're not just bringing in water, you're bringing liquid, right? That liquid can have stuff dissolved in it, right? So not just water. That's the problem with this choice. Pinocytosis increases the surface area of the plasma membrane, whereas the other one decreases the surface membrane. That's not true, because for both of them, you are bringing things in, and when you bring things in, you are taking a slice of the membrane to make it into a vesicle, right? So regardless of whether you're doing phagocytosis, pinocytosis, or receptor-mediated endocytosis, all of them will decrease the surface area of the plasma membrane. The only one that will allow you to add things to the cell membrane to make it bigger is exocytosis, which is like in a completely different categories. Tinshira asks, are we going to have any diagrams related questions? You might, right? You might have to actually, like you mean like labeling the organelles like you did in lecture? Yeah, okay, no labeling this time. Yeah, but I might show you a diagram and then ask you questions about it though. Okay, like I might show you a diagram of a chloroplast and say, you know, what process takes place in the organelle shown in the diagram. You have to know it's photosynthesis, for example. Okay. All right, let's do another one here. Select all the false statement below. These are the multi-select questions. Right? In order to get 4 marks, you must choose all the correct one and leave the incorrect ones alone. Okay, so you lose mark for missing correct answer, and you also lose mark for choosing incorrect answers. Okay, these are 2 marks each. Okay, so those are the ones you want to spend more time on to make sure you get it right. You do get part marks though. Okay, like you can get like, that's why some of you email me and say, how come my score is like 12.21 out of whatever, like where does the 0.21 came from? It comes from these multi-select question. You can get anywhere between 0 to 2 depending on how many answers you missed or included as extra. So let's see. Smooth ER is part of the endomembrane system, right? That's true. If I were making a cheat sheet, I would have a section that title endomembrane system, and then I list all the components in it, right? So there is the nuclear envelope, there is the rough smooth ER, Golgi body, and lysosome. Okay, of course you can just memorize it, but again, if you're having trouble memorizing all these facts, it's good to have a reference sheet. So that's correct. So I'm not going to choose it. I'm looking for the false one. Okay, so that's correct. Muscle cells are expected to have an abundance of mitochondria. You must have heard me said that a couple times by now, right? Muscle cells are very energy demanding. They contract, they move all the time. We need more mitochondria. That's correct. We're not going to choose it. We're looking for the false ones. So that's done. That's done. Chloroplasts are only found in plant cells. Okay? I think a lot of people choose this one as false. Sorry, many people thought this was true, but actually this is not true. Okay, this is false. So I'm gonna check it because I'm looking for the false one. The problem is only in lecture, you can check the recording. I said chloroplasts are found in plant cells, not in animal cells. But you will also find chloroplasts in other organisms, right? That lives in ponds, right? We call them protists, right? Things like the paramecium and the amoeba, those are examples of protists. So there are other ones that lives in ponds, and they might have chloroplasts in them as well. So they're not just found in plant cells. So that's the false one. All cells have cell wall to protect itself from environment. That is also false. Animal cells certainly do not have cell wall. Most people got this one. Right? When I looked at everybody's quiz, I think a lot of people missed this choice, and that's why they didn't get full mark. If you identify a cell with large amount of rough endoplasmic reticulum, which could not be a logical conclusion about the cell. Okay, so before I read any of the options, I should remind myself what the rough ER does. Rough ER makes protein, right? Okay, and it's part of the endomembrane system. That's the two pieces of information that I recall when I see rough ER. So let's take a look at this with that in mind. Large amount of protein hormones are exported by cells. Yeah, I mean that's logical. The fact that rough ER makes protein, then I can safely assume that this cell makes a lot of proteins. Digestive enzymes for export are manufactured by that cell. That is also logical because we know enzymes are proteins, right? So if this cell makes a lot of enzymes, I probably would expect it to have lots of rough ER as well. Large quantity of enzymes for biological. So again, large quantity of enzyme. That's it. I don't care what the enzyme is for, right? The later part doesn't really matter. What matters is this thing makes enzyme. If you mix a lot of enzyme, I expect it to have lots of rough ER. Steroid hormones are produced by that cell. Should I check this one? What do you guys think? Does steroid or hormone have anything to do with rough ER?
No.
No,
The
right?
SCR.
That's the SER, exactly. That's a smooth ER, so no need to choose that one. This is an animal cell. Are rough ER only found in animal cell? Can we find rough ER in plant cells, for example? Right?
Yes.
Yeah, we can find rough ER in plant cells, right? So just because it has a lot of rough ER doesn't mean it's an animal cell. It could be a plant cell. It could be a fungus cell. It could be a protist cell. It could be, whatever cell, right? So the correct answers are just the first three. Okay. Yeah, go ahead, please.
Oh, on the last one, this is an animal cell. It doesn't say it's only like it can be an animal cell, right? So how is it?
It says this is an animal cell, right? This
It is an animal cell, right?
Yeah, but it's not necessarily an animal cell, right? It's not a logical conclusion. Just because it has a lot of rough ER does not mean it is an animal cell, right? It could be a plant cell. It could be other cells, right? So I cannot conclude it is an animal cell based on this alone. That's why it's not the right answer, right? Am I understanding your question correctly or?
Yes.
Okay. When biological membranes are frozen, now this is an application question, right? You guys keep on asking me what application question looks like, right? This not something that you can look up in the notes. It's not something you can just find on the internet. You have to think about it, okay? So when biological membranes are frozen, then fractured means broken up, they tend to break along the middle of the bilayer. So like imagine your phospholipid like this. Okay, you're gonna freeze this thing and then you're gonna break it. They're saying how will it break? Well, it tends to break like right in the middle, in the bilayer like that. Okay, so this thing peels off, this thing peels off. That's what it's saying. The best explanation for this is, all right, let's see the answers. The carbon-carbon bonds of phospholipid tails are easily broken. Okay, so that's talking about the bond within the phospholipids. We're not breaking the phospholipid. It's not like I'm taking the tail and then I'm breaking off a chunk of tail, right? That's what this is talking about, the carbon-carbon bond, right? Okay, so that's not it. Interactions between the hydrophobic tails that holds the membrane together are the weakest at this point, right? That is true, right? What's holding these tails together, right? These are all hydrophobic tails. Okay, so those interactions are weak. That's why it is easiest to break down. So that's the right one. Okay, so let's take a look at the other ones and see why they're not right. Hydrophilic interaction between opposite membrane surfaces are destroyed on freezing. This head and this head are so far apart that there's no way for them to interact. There's a whole layer of hydrophobic tails in between. They're not going to interact at all, so there's nothing to destroy. The integral proteins are not strong enough to hold the bilayer membrane together. The integral proteins are the ones that goes through right there, okay? And if that's the case, if the integral proteins are breaking apart, then you should end up having holes in the membrane, and it should not like be peeling off, right? Because the integral proteins are just inserted throughout, right? So that's not the answer. Water that is present in the middle of the layer freezes and therefore easily fractured. There's no water in the middle layer. The middle layer is hydrophobic, right? So you shouldn't be able to find any water in there to begin with. Correct answer is the second one. All right. This one, we spent quite a bit of time talking about it in class, but let's go through it again. First of all, even without reading any of these answers, you have to think about which way the water will move. By now, you should have a good idea, right? Which way does the water move? To the left or to the right?
To the left.
Left.
It's going to go to the side that is more concentrated, right? So on my paper, I'm going to put an arrow so I don't forget. Next thing you're going to know, you're going to think about is if the water moves to the left, water level is going to go up here. Water level is going to go down here, right? Okay, so let's see. Concentration of solute will increase on the right side. That's true, because as you remove the water, this side gets more concentrated, right? You have the red balls are not going anywhere. You're just removing the water. When you remove the water from a solution, the solution gets more concentrated. Okay, think about like, you know, I don't know, like if you guys coconut. When you're cooking, you reduce the sauce, right? By heating it up. So the water evaporates, the sauce gets more concentrated, right? It tastes sweeter, for example, if it has sugar in it. So as the water moves from one side to the other side, it causes the water to go down and solute concentration goes up. Water level will rise on the left. That's true. Water will move towards the left side by osmosis. That is also true. Solute will diffuse towards the right side. Remember, the membrane does not allow the solute to go, right? The question says so. The membrane located at the bottom is only permeable to water. So in this case, there are 3 correct answers. All right, so the multi-selects are very difficult, I understand, right? Because you don't know how many answers you're supposed to choose, and they all seem kind of correct. But thankfully, there are only 8 of those on the test. So I hope this helps, right? You can go through these questions again on your own. I'm not saying they will show up definitely on your test, but they might. Again, it's all randomized, right? I don't know, but hopefully this address some concerns, because you guys are saying you don't know which question you got wrong, right? Anything you guys want to ask me? All right. Now, for the interest of time, I'm just going to skip to lecture 5 here, because I did review some of these things with you already with the test quiz questions. Obviously, go through the whole hydrophobic, the whole hypertonic, hypotonic, and isotonic situation. Make sure you know what goes where and the consequences, right? Those are important things to refresh yourself about. Okay, here, what is an organic molecule? Organic molecule is any molecule that contains carbon. There are a few exceptions, except for carbon dioxide, carbon monoxide, and cyanide. Whoever came up with this definition, they're like, oh, let's not include these things. Not sure why, but that's the way it is. List all products of glycolysis. Again, if you are doing the help sheet for the test, I definitely would have one that lists all the major steps. So glycolysis, pyruvate, oxidation, Krebs cycle, etc. And I will have all the major products listed. Okay. So very quickly, right, for glycolysis, we have 2 net ATP, gonna have 2 NADH, and we have 2 pyruvate, right? I believe we did that last class at the beginning anyway. That there's a chart like this. Okay. So I know I sound like a broken record, but there is a difference between you making this chart on your help sheet as opposed to you're like, oh, I have this in the study guide anyways, right? In the answer key, I can just look it up when I need to. The thing is, when it's in the study guide, like it's like all over the place, right? Like it takes time to find it, right? So that's why people are saying you run out of time. Okay? Maybe some of it is because you have to read the question many times. Maybe sometimes you spend too much time looking for information that you need to know. And again, I'm not saying you're not supposed to look up information. By all means, go look up the information. It's fine. It's not cheating. Okay, it's only cheating when you try to copy answers from the internet and put it in the written questions, right? That it's not acceptable. But if you have to look up information from the notes, that's fine, right? So rather than having it scatter throughout your binder, put it all on the same page, okay? And when you take the time to create that sheet, you will know exactly where things are, right? Okay, so it will save you some time. Okay, so something like this is definitely important, right? Something like this is also quite important to have, right? All right, let's go back to here. In cellular respiration, glucose is oxidized while oxygen is reduced. We talked about this multiple times already. You should know, in general, not just glucose, right? If anything is oxidized, it means it loses electron. If anything is reduced, it gains electron. Okay? So, glucose is going to be oxidized into carbon dioxide. Water will be reduced into oxygen. NAD+ will be reduced into NADH. FAD will be reduced into FADH2. Right? These are all examples of reduction. Okay. And then over here, right, I can write the other way around. NADH is going to get oxidized into NAD+. FADH2 is going to get oxidized into FAD. Right, so these are oxidation. Okay. Again, something useful to have if you confuse between the two, to have on your help sheet. Okay, what is the role of NADH and FADH2 in cellular respiration? They are there to capture high energy electron from step 1 to 3, right? Okay, glycolysis, pyruvate oxidation, and Krebs cycle, and deliver them to ETC. That's the job. They are the taxi. They are the shuttle that goes back and forth, right? Significance of adding coenzyme A to pyruvate during pyruvate oxidation. This is to re-energize the molecule such that it will react further in the Krebs cycle. Right, I keep on saying in class, by the time the glucose has gone through glycolysis, it's already low in potential energy, right? Because you produce some ATP, you remove some high energy electron. So you need to recharge the molecule. You need to do that by adding the coenzyme A. All right, so here we have some math questions. You will need your calculator, okay, for the test. There will be some calculations involved, something similar to this. If there are 50 NADH, 19 FADH2, how many ATPs are we going to be able to produce? So in class, we already said 1 NADH under ideal condition, which is why it's asking for maximum, right? Ideal condition, we can times this by 3. We'll get 150. And for FADH2, what do I multiply this by? 2, that's right. So that's going to give you 38, right? And then you just add them up, which is 188. That's the correct answer. Okay, the inner mitochondrial membrane is about 5 times the area of the outer membrane. What is the purpose of this? Well, the ETC is embedded in the inner membrane. Right? More surface area equals to more ETC, which equals to more ATP. Remember, the ETC is where we will harness the electron's energy to generate the H plus gradient. When the H plus goes back into the matrix, that's how you're going to produce the ATP. So if you have more ETC available, if you can pack more ETC into the inner membrane, you will be able to create more ATP. Right? So that's why you have a big surface area for the inner membrane. There's no ETC in the outer membrane. I don't need more area for the outer membrane. Nothing's going on there. Okay. Which step of cellular respiration generates the greatest amount of ATP? That's ETC, right? Okay. Remember, we generate ATP using substrate level phosphorylation. That only accounts for 11% of the ATPs. And then we generate using the oxidative phosphorylation, right? That's all the ATPs that are generated during the ETC, right? 27 of them, right? 32 of them, sorry. Okay. 32 ATP. Describe what happens to the H plus. I told you already, right? They come back in and generate ATP. And what is the purpose of the final electron acceptor? That is for oxygen. So first of all, final electron acceptor is the O2. So oxygen removes electrons from ETC, right? So that to allow continuous electron flow. If there's nothing there to remove the electron, the whole ETC will get clogged up and you won't be able to deposit additional electrons, and you won't be able to pump H plus, and then everything stops. No ATP production. Okay. Now in class, we talk about different kinds of poison, all right? We talk about the rotenone, we talk about DNP, making holes in the inner membrane. You want to review those as well. You want to see what kind of problem it causes. I could ask you, the poison DNP interferes with ATP production in what ways? Okay, and then you have to know, oh, DNP makes holes, make the inner mitochondrial membrane leaky. Okay, there are other poisons, right? Like cyanide, right? For example, oligomycin, right? Okay, so just review those slides and make sure you know what the poison does. Okay. I know a lot is a lot of information, okay, but you still have 2 more days to prepare for it. If you again need help, feel free to email me. Some of you email me and also post it on the Padlet and ask me if I'm still going to be on campus this week. I won't be, but I will be available for extra help if you need to. Okay, if you want to meet with me like through Zoom in a private meeting or in small groups, we can set that up. I have some time tomorrow morning. Otherwise, just email me. Okay, I'll be happy to answer your questions. Okay, so this is my last week for the summer semester. As I told you, Professor Maren Airy, he will take over next week. He will probably be posting some information on eCentennial regarding his session later on this week. So please stay tuned. He will have a new schedule and he will tell you how he runs his class and things like that. Okay, so I wish you all the best for the rest of your semester. And thank you for having me as your teacher for the first 5 weeks of the course. Okay, and all the best with the test on Wednesday. Okay, take care, everybody. Bye-bye.
Thank you. Take care.
Thank you. Thank you.
Thank you.
Thank you.